Welcome to Gene Silencing. I'm Dr. Shay, and I'm excited to share with everyone what our stream does to silence genes. There are two main questions. How does the environment affect our gene expression patterns? And how would that affect our descendants? We know that the environment, such as changes in diet, stress levels, toxins, and experiences, could affect our genes. Overall, changes that impact our ancestors could have long-lasting, unknown effects in descendants. A change in gene expression refers to whether the gene is on or off, meaning there are either more or less molecules of the gene product. We are all derived from a single cell, which contains the genome, made up of the DNA sequence, and the epigenome, made up of intricate networks of regulatory molecules. Defining these differences is what separates genetics from epigenetics. In this stream, we will be working with elegant worms to understand heritable epigenetic effects. C. elegans is an excellent model to use because they are transparent, which allows you to see different gene expression patterns. They are hermaphroditic, so they're easy to maintain in the lab. They reproduce and develop quickly, and there are numerous tools and resources available for these tiny creatures. Furthermore, we can silence genes at will by delivering double-stranded RNA into the worms either by microinjections or exposing them to bacteria expressing double-stranded RNA. Andrew Fire and Craig Mello won the 2006 Nobel Prize in Physiology or Medicine for this discovery. This pathway of epigenetic gene silencing is often referred to as the RNA interference pathway, or RNAi pathway. Double-stranded RNA can trigger a cascade of silencing effects by targeting the mRNA product of complementary sequence. To maintain silencing across generations, this process is dependent on a positive feedback loop consisting of regulatory molecules such as RDE3, HRDE1, and small interfering RNAs called PUG RNAs and 22G RNAs, to name a few. This can result in both co-transcriptional and post-transcriptional silencing. In C. elegans, a new phenomena of epigenetic gene silencing was discovered. Mating could trigger hundreds of generations of silencing. This phenomena, referred to as mating-induced silencing, was discovered in Dr. Anthony Joseph's lab at the University of Maryland. They showed that this single-copy transgene called T and a variant of it called T-cherry are only susceptible to silencing when males, expressing the gene, mate with hermaphrodites that lack the gene. This silencing is stable across generations despite the initial trigger. The University of Maryland's News and Events Digest wrote an article about it titling Match Matters. The right combination of parents can turn a gene off indefinitely. If you'd like to learn more, check out the link below. Like dsRNA-induced silencing, there's a similar positive feedback loop required for maintaining silencing across generations. However, mating triggers another class of small interfering RNAs called piRNAs that are necessary to start this cycle. It's unclear what other regulators are involved in this pathway, and further research is necessary to understand the mechanisms of mating-induced silencing. This brings us to our research goals. What regulators and interactions of this gene sequence make a gene susceptible or resistant to stable epigenetic gene silencing, triggered, for example, by mating? In our stream, we will be manipulating the sequences of this gene containing the fluorescent reporter gene mCherry, specifically editing the promoter and 3' UTR because of its known regulatory properties of gene expression. Hypothetically, the promoter could recruit positive regulators that promote gene expression, whereas the 3' UTR could recruit negative regulators that promote gene silencing. Our first steps will be to edit the promoter and 3' UTR using several tools and techniques in molecular biology and genetics. These include, but not limited to, using a plasmid editor software called APE, genome editing tool called CRISPR, methods for genotyping such as PCR and gel electrophoresis, sequencing, fluorescent microscopy, and a simulating platform called NetLoco. Thank you all for your time and attention. Additional resources can be found in the links below, and I hope you enjoyed learning about the Fire gene silencing stream.